Welcome, everybody. It is my distinct pleasure to host Professor Eshel Ben Jacob and Zan Gill, who helped us arrange this. I mean, Zan is our friend for a long time. She runs the Microbes and Mind Forum, which I do recommend that you look it up using your favorite search engine. And you will find a lot of interesting information on her website.
Well, we are really fortunate to have Professor Eshel Ben Jacob here from Tel Aviv University, where he holds the McGee Glass Chair of Physics, and as past president of the Israel Physical Society. The significance of his work is that he has uncovered basic principles in physics that have a broad range of applications. So he's not only co-developed the first hybrid neural memory chip, which was named by Scientific American in 2007 as one of the 50 most important achievements in all fields of science and technology for that year. His principles have been applied in such diverse areas as neural networks in the brain, the immune system, search, next generation search via social networks, and stock market volatility and collapse. So we're really looking forward to what he has to say and to your questions. And please put your comments also on the sign up as it goes around.
Okay, thank you. Morning to everybody. It's not going to be a formal talk, so please ask questions during the lecture. So let's go. Okay, so what you see, and I'll take you through some observations and let your own thinking tell you what you think you see, and then I'll give you some hints to convince you that the bacteria are intelligent, and not just that they're intelligent, but we can learn from their social intelligence about many things, including maybe things that people here in Google are doing. Okay, so let's go on. Okay, so just introduction, and people like to think about themselves. So the first thing is to show you something that we can define as clashes of rudimentary intelligence. What you're going to see here happens in this very moment in the body of each and every one of you. These are red blood cells. This is a white blood cell. This is a bacterium. The white blood cell wants to eat the bacterium would like to survive. So now you see what happens and try to see that it runs away, but try to figure out if it really runs away, it knows what it's doing, or it's just making a random walk. Okay? To help you, there will be another bacterium that will be here on the side, which is not chased, and then look what it does. Okay? So let's play the movie. The music is not going inside your body now. See this one here, and the one you just chased. Okay. Sorry about that. I forgot that there are some words that are not supposed to be mentioned on the YouTube, but since this movie is from YouTube, I guess it's okay. Now, what you saw here, that it's running away, the bacterium, it's funny, but it's very serious, because it means that the bacterium is able to sense chemicals or sometimes even ripples formed by the white blood cell, keep memory of what happens before, process the information, and react accordingly. If you notice, from time to time, it makes 90 degrees angle, run away. At the same time, the white blood cell has to be able to sense chemicals secreted by the bacterium, have memory, process the information, and react accordingly. And you can play games like this. When I say game, it requires some work to model the motion of the white blood cells. I don't have time to get into it, but some movies like this that they're chasing. And you see, this is the case that eventually it will win. Some of you might hear that one of the leading health problems in the world is the fact that we have multiple drug resistance bacteria. And this is one of the highest risks on our health. This is on one hand. On the other hand, bacteria are also our best friends. Some of you don't know, although here you have some nice food, healthy food, but bacteria in our guts process eventually the food. We would not be able to live without the bacteria in our guts. They also communicate with our brain. They decompose all our waste products, and so on and so forth. But let's talk about the risk of bacteria. And what we realized in last years is that bacteria strike back. After the era that we saw that with antibiotics, we can get rid of all the bacteria-caused diseases, all of a sudden, these diseases reappear, and the death rate in hospitals in the West from bacteria is number 3 in the West. Okay, so how come? We just saw how the white blood cell won. How can it be? Apparently, we overlooked something when we treated bacteria. And what we did is a basic mistake that people do when they have clashes. We undermine the capabilities of our opponent. And what I try to convince you is that bacteria form social networks. Not only one colony form a big social network, but different colonies of different species form social networks, and they form social network long before Google appeared. They form social networks all over the globe. When we develop antibiotics, it takes us about 5-7 years, hundreds of millions of dollars. The bacteria develop the resistance within a year or two, and they share the resistance in cassettes, genetic cassettes called plasmids, to other bacteria all over the globe within a year or two. Okay, so let's just introduce you to bacteria and the wisdom of the colony. What you see here, as I mentioned before, it's supposed to be a circle. Okay, this is real life. The pattern is the real pattern. The colors are artistic colors that I changed or translated the colors from blue to dark blue to light blue to red and white. These are high concentration, and the red is lower concentration. Inoculate the bacteria here. This is a strain that I discovered, and then they expand outward. This strain is specialized in moving on very hard surfaces. What you see here are many things that discussed here, but to give you a real idea or to shock you is that this is about 14 centimeter or 6 inches. The size of a bacterium is about 1 micron. The number of bacteria cells here is about 100 times the number of people on Earth. And each one is both an actor and a spectator in this big global village, which is manifold larger than the number of people on Earth. And they all know what they're doing. You see how symmetric it is. They send these pioneering parties that I'll show you in a second. These pioneering parties are also the production organ of the bacteria. They are bacteria that turn around like merry-goes around, and this way they pave the way for the rest of the colony to expand. And let's zoom in at the beginning when they just start. You see each bar here is one bacterium. The rate is real rate, and the magnifications is 500 time magnification. This is from a movie I have to mention that I made for the BBC, so I can take extract from it, and some of the sound that you learn here, it's not my sound, but of an English lady. Go up.
Their ability to organize and adapt to change explains why bacteria threaten our health, even when we use antibiotics against them. It also explains why they've made such a good job of colonizing the rest of the world, from deep in the Earth's crust to the edge of space, maybe even beyond. Bacteria are smart bugs, all right.
The last part that you saw here is not real time, but is accelerated 1 times 60, 1 to 60. Okay, this is how they go, the same bacteria, on a very hard surface, so they have to work as a group and pave the way. If you go them on a surface which is not so hard, this bacteria I forgot to mention, they are also lubricating bacteria. In order to move on the surface, they have to secrete lubricants that enables them to swim together. And that's, yeah.
Do they rotate differently in the north and the southern hemisphere?
Good question. These are not, they form, the rotation is innate rotation. It's not due to the magnetic field on Earth. There are some magnetotactic bacteria that are sensitive to the magnetic field, and they behave differently in the north and the south hemisphere. Okay, here. Okay, when you go them on a surface which is not so hard, what they do, instead of having these groups, pioneering group, that pave the way, they send swarms, okay? I'm sure that many here, because we are in California, are marathon runners, so I'll give you an idea of this swarm that you'll see what happens. Okay, so this is a swarm. To get a perspective to human being, each dot is one bacterium. So think about it, that this is a swarm of a group of thousands or hundreds of thousands of marathon runners, because they move quite fast. They move at a speed of about 10 seconds for 100 meters. In order to move, they have to pave the way. So all these marathon runners take buckets of oil, maybe scatter, I should say, that they put down and they move. That's what keeps them together. This is just a free motion that I show you. And then I show you what happens when they find food in the vicinity. Okay? So look at the motion. The motion is quite complex. You see some come here, go back, come from here and go back, and this huge swarm is moving on. Okay? Now let's see if they're really smart. What does it mean, really smart? Let's go to the next thing. Now they move along, and what I do, I add here a food source. Think about it, that each one of the marathon runners is also blind. You cannot see that there is a pizza stand about, let's say, 5 miles away. Okay? What they can do is smell the pizza in its local location and calculate the changes in the smell and communicate with the other guy. Now, so this is the swarm moving, and you'll see what happened. Now, for you to be able to think freely, as they say, I tell you that in order to do what they do, you must have collective or distributed information processing and social networking by chemical tweeting. Okay? So now let's see what they do. So you add this in, okay? So the food diffuses, then look at it. They move, takes them some time. They don't carry laptops with them, so it takes them some time to do the processing. Okay, and they go and slow, realize something is going on, and then collectively, whoop, they go to the food. Now think about it. I don't know here in the US, but in Israel, if we'll have a group like this of marathon runners, there will be pizza, and they smell the pizza, they all rush over. And there will be a big mess. Here they have to coordinate, because in order to make this turn, the bacteria over here have to communicate with the bacteria over here, and all of them, the ones which are further away, have to run faster, so the entire swarm will turn. Yeah?
Is there do you see any type of leaders per se, or is it just a group?
Okay, that's, again, excellent question. One of the special things about the bacteria, and that's maybe why it is so important for social networking that we see right now in the world. Human beings now try to imitate the bacteria. There is no leader. They all change, but we'll see later there is a distribution of tasks. So each one can play every role, but when they play a role, there is a distribution of tasks, cell differentiation, and they maintain it over some time. But there is no pre-born leader or elected leader. Although there are some bacteria in some conditions that are differentiate between adventurous and social. Those that like to take risk and those that try to stay back. Yeah?
Are there communication hubs and spokes at all, or is it completely just amorphous?
Okay. There are communication hubs that in the picture that I showed before, that you have these centers. Some of the center is communication hub. They are less active in looking for food, eating, and reproducing. So they invest less in reproduction and more in communication. They are the older one that stay there in the colony at the beginning, and they send messages to the kids how to behave themselves. And if they get messages over there, there is antibiotic, what they do, they tell the colony, move over there. But we'll come to it. Okay, thanks for the question. Very important. Yeah. I don't remember who was. Yeah.
Do you know what chemical they secreted to do the communication?
In some cases we know. In many cases we do not. I'll mention it along the lecture further on.
What defines the boundary of the swarm?
What?
What defines the boundary?
The boundary, because what defines the boundary is they secrete lubricant. And this is, again, I show. They are smart enough to secrete the lubricant that will be sufficiently viscous so it will form a boundary. Okay, so let's move on. Now that we decided when I was young, I was interested in philosophy, so I knew about this dilemma of choice. It is very popular that you give a donkey 2 pieces of food, not spinach, and then he cannot decide which one to eat, and he dies from hunger. We try to imitate the same thing for bacteria. Now, it sounds like a simple experiment relative to the high-tech experiments that people do nowadays, but it is not. When you have a swarm like this moving under the microscope, you want to put 2 pieces of food at the right distance, the right amount. It's hard. Sometimes the kitchen looks a bit messy. And luckily so, because what we discover is something that we didn't expect. So you have 2 pieces of food here, and then we dropped something here, and a little pieces here. And what we expected to see, that the swarm will go over here, and then go over here. Instead, the bacteria turn out to be smarter. So first, they behave like we expected, but then there was a change. They even noticed this little piece here, and they noticed this little piece far away, and look what they do. Good, yeah. This is something, one of the most exciting things I ever saw. And we spent and looked at it and looked at it and looked at it. We now feel that we have some ideas how, I cannot say how the hell they do it. Just to show you again in full glory. And then you see that they slow and digest the food. Slow down after they have the food, like an animal that swallows this thing. Okay, these are the messages. Bacteria are smart and communicating beasts, unlike people thought before. Advanced communication. The communication is very advanced. I show you. Those of you that will live, later on I'll show that for one type of decision, they use 12 different messages, and they form messages, combination of them, that are far more complex than the Twitter messages, I had coffee, or the coffee was good, or other type of Twitter messages of 140 characters. If you think about each of the chemical has a quite complex structure, and you have all the combination of the chemicals or the messages. I say tweeting, but it's far more advanced than the usual Twitter messages. And it is important that the communication, and that was very hard to convey, and that has to do with social networking, that the communication convey semantic messages, meaning, not only information. And they carry dialogue using the communication, and eventually they can make decisions in a collective way. And this is essential, the communication, for their social intelligence. And we'll see distribution of tasks, distributed information processing, learning from experience, planning for the future. What a surprise. Unlike human beings, societies, they do plan for the future. If you give them a lot of food, Some people say they are stupid. Some people say they are smart. They don't use all the resources and exhaust all the resources because they know that things can change. So they keep things and save for the future. In a population, they always keep different type of different expertise, different sectors if you compare it to society, so that if conditions will change, they will have some bacteria that already expert in facing the new conditions. Okay, so they always keep this, and they can make collective decision, and they have rapid collective adaptability. Okay, take-home message 2 is the implication, is the power of social network. I'll show you the power of cooperation, self-organization, and I'll show you example of really what we see now in society, that the social networking of the bacteria can lead to collective identity switch. The bacteria can really, the entire colony, can change its identity by changing the identity, those of you with background in biology, epigenetic changes in individual bacteria. Okay, so they really change themselves in a meaningful way. And again, the question that I asked before, without a leader. Okay, just they read the condition, they communicate, and they do it. And there are also many applications. There are new strategies to fight bacteria that we showed. Once you understand that they are smart, you understand that in order to fight bacteria, you have to outsmart them. And you find new strategies also to make the use of bacteria in our guts. You can start to understand why eating some substance is good for you and some is not as good for you. Okay? This is something which is very important, our application. But new strategies to fight cancer is something recent that we do. Right now, what happened in society, Western society and other societies, in terms of cancer, the situation is very reminiscent what it was with bacteria. People think that cancer cells are defective normal cells with limited capabilities. Because they are mutations, they are defectives. So we did not succeed to fight cancer because we undermined their abilities. We now see many parallelism between tumor and bacteria. We understand that they form meta-communities. They have very sophisticated communication, cooperation, decision-making, and so on. And I'll reflect on it a little in future, later on. New game theory. I'll show you that when they do decision, they have smarter game theory than the game theory that usually used in mathematics. And new robots. Show you how we can do. Some people in NASA, other places are interested in robots now to explore new terrain. And I'll show you how we can use some idea of distributed new robots that will show some swarming intelligence and eventually new generation of cell networks. So this will be the topics, just the structure. Now that we know where we're heading, let's talk about simple example. This is a bit different strain of bacteria. So this is a very simple example because what I do here, I give them a problem. The problem is that this bacteria also, it's another strain that I develop. In order to move, they need high density to secrete the fluid, the lubricant. But I don't give them enough food. So what they do, they make this branching pattern. Okay? Why branching? Because within a branch, the density is high enough so they can move together. But if you average over, the density of bacteria fits the amount of food that we have. That's a simple thing. If you look at here inside, you see that they're swimming. The swimming itself is quite fantastic. This is real time, 500 magnification, and you see how they swim. Now, all these things, you look at it, and you say, OK, they're swimming. What a big deal. They're swimming. Well, let's look a bit closer, just on this element to share with you the many things that we can learn. They swim by flagella. Look at this. This is 1 micron, and this is about, can be 30 microns. So think about it. if I translate it to human being, that you are 1 meter, between 1 meter and 2 meters, okay? And you have a tail of about 50 meters, okay? And you move with such a tail by turning it around. You move like this, okay? Now, think about it, that each of you has a tail of about 40, 50 meters, and now you have to go like this somewhere. What will happen? In 1 second, all these tails will be big and tangled, and you'll not move anywhere. Okay? But somehow, you see, this is a low density, so that you can see the tails, but they can swim in a high density and still manage to do it. We don't know how to solve this hydrodynamic problem. But we know, and this is important, that they move the tail for a few seconds, and then they unwind the tail, they turn around, and pick a new direction random. This is how they do random walk. Okay. So this is the thing. Okay, so this is in low density how they do it, but they do it in high density, as we saw before. Okay, mathematical ingenuity 1. Why mathematical ingenuity? Because if you would secrete lubricant in order to move on a hard surface, you'd think that you need to secrete a low viscosity lubricant so you can swim better. No, but if you secrete low viscosity, the branches will not be well defined. And they have to set the level of the viscosity of the lubricant to be exactly solve this optimization problem, that within a branch, the density will just be right, and the distance between the branches will just be right. Yes?
You mentioned, I think, the velocity is like 10 microns per minute, per second.
The other bacteria. In this one, it's more like 1 micron per 1 second.
So they're able to move their whole body length, basically, for their body, 1 micron typically per second. Equivalent for us, that would be extremely fast.
Yeah, that's what I mentioned. This is running marathon extremely fast. Extremely fast.
So how are they energy regulation?
Okay, we'll come back to it later, okay, because it will take me on a different direction. But remember the question and, oh, okay. So, but that's not all they can do. Well, I skipped this one. This is just to share with you that we also do equations. But we don't want to do, you believe me. Now, if you lower the level of nutrient, at high nutrient, they have something dense. Low nutrient, you have something fractal like this. And then there is a surprise. When you go to very low nutrient, again, it's dense and very thin branches. So this is another challenge that we tried to understand for a long time. And then we figure out that they have a trick to do it. And the trick is very important to share with you, and this is called chemotaxis. Chemotaxis is very important because it was invented by the bacteria, but it's used throughout the kingdom of all animals. And that's why it is nice to learn about it. In the case of swimming bacteria, what happens? Let's say that they have food over there. And we saw that they do random walk. And they want to bias the walk over there. So how do they do it? What they do, remember, they go like this with this tumble. So they make a step and measure the level of smell, if you want. The bacteria measure the concentration of some chemical. And then they make another step and measure it. And then they calculate the difference. If they see that the change in concentration goes up, they make longer steps in this direction before they tumble around. Pick a new direction, measure. If it goes down, they tumble before. So on average, they have a drift towards the direction of high concentration. This is called attractive. They have repulsive, which is the opposite. If they see that it goes up, they make shorter steps. Okay. What we discovered in the early 90s is that the bacteria use this to do chemotactic signaling. Namely, they secrete chemicals that cause their peers to either move towards them or to move away from them. And I'll show you an example of what I mean. They have good times, they have plenty of food, they send chemicals that tell their peer come over. Okay? Help me is the feast and clean the dishes afterwards. If they don't have enough food or encounter some stress, they send chemicals that tell the peers, move away. So there are two elements. There are additional type of chemotaxis. There is a chemotaxis which acts like a Swiss or French cheese, that if you are from a distance, it smells good to you. You get closer, you smell it, and you're repelled. Okay? So there are all kind of combination of the two things. But this is a chemotaxis. Why it's so important? This is the way that our brain, the neurons, wire together. How the neurons send processes to connect to other neurons. They use chemotaxis. Not only do they use chemotaxis, the agents are very similar to those that have been invented by the bacteria. The way that we reproduce, the sperm swim and navigates toward the egg, it's the same thing. They use chemotaxis in order to do the navigation. Most butterfly moves towards the female, the male most butterfly. It also used by chemotaxis. The female secretes some pheromones, and they move like this towards the female. So this is something which is used all over the chemotaxis. The bacteria use it in order to generate the patterns, and if you include it in the model, indeed what you get is agreement with the results. Okay. Now let's look at some implications. So I show you this bacteria, and I told you that we have some implication to social networking. So I showed you that they can change the pattern of the colony, fit it, adjust it, and so on and so forth. But there are some times that despite of all these things, the condition changed so dramatically. Okay? For example, the surface is not as hard as it was before. It is a time to a more major change in the pattern of the behavior. Instead of acting individually, like random walk that I showed you before, now, because the condition changed, they decide to act in a more organized fashion and make identity change. And look how bacteria do it. So this is the colony that I showed you, branching, simple branching. When we grow the same colony, start to grow it on condition that the surface is not as hard, or that you put obstacles, some mechanical obstacles or other obstacles. You can use imagination to think how it is translated to human being situation that we see around or have been observed right now in society. What you see, boom. Change in identity. All of a sudden, you have something which is completely different. If you look closer, you see that the individual bacterium become longer, and they swim in a much more coordinated fashion. This is how they can go around obstacles and so on. Show you another example. Different condition, it looks a different way. Sometimes it's one side, sometimes you can see many bursts of all over with this identity change, right? Like Tunisia, Egypt, okay? And it's the same type of identity switch. You see, it's a day, time, because in this case, it is imprinted, the capability of being different character is imprinted in the genome. But you have to go through the genetic chain. Not only they become longer, the bacteria, they have multiple copies of the chromosome and many other things. If you take this bacteria now from here and inoculate, you see that under this condition, you see how they look like. You see how different they are. The branches are thinner. They have this handedness, breaking symmetry. And in order to have this handedness, by the way, help them to go around obstacle, and they act in a more coordinated way. You can see this is under different condition, the same strain. And this is transition back from this type. If you take this type and put it on a very hard surface, they can make the transition back to the other pattern. Okay? So this is one thing, social revolutions by chemical tweeting. Just passing by, I could not resist to share with you the patterns are so beautiful. If you didn't notice that there is another angle to the research second, that you can turn it into art, but now the aspect ratio changed on me. So, but it's still beautiful. Yeah?
So are these collective identity switches reversible if you go back to the original
Yeah,
conditions?
yeah. It's not only original. You have, it does not go back the identity on the same condition. There are some set of conditions where you change from one type to the other, and another set of conditions that you change from here to there.
So what I'm looking for here is any kind of collective memory.
Right, epigenetic. And that's very important also for cancer cells, because now we give chemotherapy, for example, or other things. They change identity, and then they appear in a different form, and many other types. So it goes all over, many applications, this idea. And we discovered it in bacteria. It was not known before. About art, it's those of you that are interested in art and walking here around with a guard. I noticed that people are interested in design, so I'll show you something. Let me just show you this, and I'll take the question. You see, this is a black and white color. If you color it, change the colors, you both have artistic thing into it, but it also helps us in the science because we bring to our intuition or attention some motifs that become more pronounced, and then we gives us a hint how to look for other things. So the art or coloring the colony can be also very useful in terms of the science, and look how different it is when you change the colors. Boof. See, it's a very different perception. You can see these things which are, you can see it yourself and decide. Yes?
So are those the same bacteria, or are they new ones?
These are the same bacteria. The ones that, the question was if they are the same bacteria. Yeah. And that brings back what is the same. You know, what does it mean, the same bacteria? It's the same genome, but the genome goes through rearrangement and some changes. The fact that the question before was that you can bring it back to the previous version means that they are the same and it's not a mutation. Okay? It's like cell differentiation in the body for some sense, that you in principle can take a skin cell or skin stem cell and make it into another cell. Okay? Yeah.
So what will happen if the whole bacteria has different sections? So let's say one section made a switch, but it dies because it was too hostile or the switch wasn't effective.
All right. So
what will happen with this kind of section?
okay. So this is we get into the details, and you are right. I'll try to get back to it later on. Then before they make the switch, they probably explore these things. And I show another example of making a decision-making between different things, but they do make a switch sometimes and die. And the ones that make the switch will not survive. Okay? They also, I didn't reflect on the fact that once you have cooperation, you have also ones that are defectors, that make a switch in order to take advantage of the other ones. And they are successful unless the condition change, and then they are less successful than the original one. So it's a lot of going on. Yeah, sure. Yeah.
To what extent is the underlying mechanism here of adaptation epigenomic as opposed to pure gene expression?
Okay, it's epigenetic.
Epigenetic, okay.
Yeah, it is epigenetic. The change is not just gene expression, because it's not reversible like gene expression, it's epigenetic. It's much more than expression. Okay. Now let's go to social intelligence of the first strain that I showed you. This is a modular organization, and I use this strain in order to show some of the notions that we talked about. Just to show you each dot here, it's a group of bacteria like this. You see that they turn around. This is a real speed, magnification 500. To understand how they do it, if you look at this movie, and you see these pictures, they both hold hands. They have the flagella, and they have polymers that extend around that they hold hands, somewhat like these skaters that make this sound, but it's more complicated because unlike the hands of human beings, these hands can be pulled like in the science fiction movies. The hands can become long and short. They are more elastic. so they can manipulate very nicely. They have additional mathematical ingenuity that bacteria close to the center goes through what is called press spore. I hope I'll get time to talk about it, what is press spore, later on, and it makes the colony more stable. This is something about the bacterial linguistic communication and social intelligence, the different patterns. Why they form the shape, we now understand that the complex shape reflects the fact that you have different variety and this high complexity. It's true in general, network and other system. If you have a complex system which not the elements are not all the same and they can change their character, the network as a whole is much more adaptable. If you have changes in the environment, you have stress, they can easily change the overall organization. If you have a society that everybody is the same, the society is very rigid and is prone to some systemic collapses. Or if you have economy, everything is the same and all the investors behave the same way, the system is prone to systemic collapses. Very important to maintain variability. And in order to maintain variability, you need a communication. So here is an example. You take this colony, grow an antibiotic, you see that they change the organization immediately. They form higher, larger vortices like this, pioneering group, and they run away from the antibiotics. See how they change the organization. Then they have learned from experience after you grow them in the presence of antibiotic, they already can grow better in the presence of antibiotic and make larger colonies. So this is another direction to explain how bacteria develop resistance to antibiotic. If you maintain them, expose them all the time to antibiotic, they collectively develop resistance, which has been ignored by human beings for about 50, 60 years. People asked before, this is to show that this is epigenetic that you ask, or that there is really distribution of tasks and cell differentiation. If you take cells from here, from the center of the colony, inoculate on a plate, you get a growth like this. You see two experiments, it looks the same. If you take bacteria from here and inoculate, they develop different pattern, which shows you that there is really a differentiation and they remember what was their task. But still, if you grow them under neutral condition, they can go back from here to the same and from here to be the same. Yes?
Can a bacteria sense something over the range of the size of a colony?
Yes, yeah. They can measure signals that are sent from all over 30 centimeters, and they can sense the signal. Okay, let's run faster. Okay, so the question that you want and the decision making is to be or not to be. Under very strong stress, starvation, the bacteria have two options. One option is to become spore, to generate spore. What does it mean? They replicate the DNA, wrap it in a very special membrane, and break open until better times will arrive. This is one of the risks of bacteria and one of the great inventions of bacteria because these spores are very resistant. In 96, I think, they took spore of bacteria to outer space on a mission, exposed them to outer space with no special clothes, brought them back to Earth, and they germinated. But some bacteria do not form spore. These are the more adventurous ones. What do? They do the opposite. They become longer, make the membrane permeable, so they can absorb food which is released by the bacteria that generate spore. Because those that generate spore make the spore, and the rest of the bacteria break open with all the pieces of DNA and the food and everything. So you have here a game theory. You have to decide, what should I do? Be safe, become spore, wait for the future, can be the future 10,000 years or whatever, or take advantage of the fact that the other ones chicken out and became spore. Now, clearly, you don't want to become competent if you don't have enough that becomes spore. So there is a game theory, and it's a collective game theory. It's not between two, it's between a number which is very large. Now, those that become spore want to be collectively. They want to form the spore together, and the reason is that when they germinate, they immediately start with a large society. So they are more or less vulnerable to some hazards in the environment. So this is a game theory. And how do you figure it out? And this was a great challenge. This is closer picture. Very nice. So you have the challenge of collective decision-making. The game theory that we have right now tells us about decision-making when you have two individuals. Should I cooperate or not cooperate? And I get 2 years in jail, 5 in jails. Big deal. Here we're talking about game theory that either I die or I don't die. Okay? I die now, or I have to wait, whoever knows how long time in the future for condition to be better. And you have to do it collectively. How do you do it? And we found that this was, or many people found that this is very essential. People try to understand decision-making on human being, but it's very hard to study decision-making of human being because it's all subjective and it's very hard to know what you do. Here, it's supposed to be simpler in the bacteria because we know all the genes. So let's talk about decision-making at the time that we have, and then I'll show you some application. So first of all, if you think about it, what does it mean to make decision? First of all, you need to decide between 2 options at least, or more. Okay, the other thing is that you need a switch, stochastic switch, because if there is something that tells you from the outside that you are in this state or at this state, if you are a transistor and some signal tells you that you are at 1 volt or 0 volt, it's not a decision. Although if you look out there, you see some articles talking about decision-making of a transistor. So you have some stochasticity, some stochastic switch. And they have it, the bacteria. Now, if you want it to be collective, this stochastic switch means that you have, when it's a specific gene, that it's self-activator, when the level goes above some level, the bacterium change into another state. Now, you want that the probability to switch will depend on the cell density, right? Because you want that if you have higher density of cells, they are more likely that some other will go through sporulation, so it makes sense to go to competence, if not. So that's something which is very well. Now, in order for this to be decision and not just stochastic switch, you also have to gate the switch. So you open the switch. It's not that you make it go to one state or another, but you open the probability to go from one state to the other at specific times. Okay, so you have to gate it, all the time inhibit it. This is a sign in gene network of inhibiting. Gate it by something. So what do you gate it by? And we figure out for the bacteria, it's very nice. You gate it by adjustable timer. It's another gene that accumulates some protein. And the way it accumulates the protein is according to the stress. And the higher the stress, this timer goes fast. And only at some levels of this timer, it relieves the gating of the switch. So these are the 2 basic elements. And the rest of it, you want the stress to affect this thing, and you want higher stress, higher rate. So what you want is a competition between 2 regulators, adjustable timer and stochastic switch. If you have high stress, you know that you don't want to take risk and go to competence because you might be dead. And you don't care if other people go or other bacteria go to sporulation and so on. You want to make a decision, go to sporulation. So that's the picture, the main picture. Now, because it's a collective, you also have cell-cell communication. And the cell-cell communication is relatively simple. You send message to the other one, what is your intention to go to sporulation? What are your intention to go to competence? You get messages for the other one, and then you regulate the cell density only according to the number of those that want to go to sporulation. So it's not important only how cells there are, but how much they incline to go to sporulation. So that's the picture of how you do it. I don't get into the detail, but this is what we figure out. And one more thing is the inhibition of inhibition principle. I'll go through fast. The decision circuit, you have combination between the gating, and the way that you do the gating is such a way that you give yourself a window of frustration. Think about yourself when you make a decision to go out of the lecture right now or not. Okay, so it's boring, so you incline more to go out, but maybe something will be interesting, so maybe I should stay. There are people out there. Reminds you that there is lunchtime. Okay, so all these things have to do with a gate, a circuit, which is very, very simple. It will look for you because you are not used to gene network, something complicated. But all that it does is you gate, you inhibit a switch, and then this one inhibit the inhibitor. Think about it. If you want to do something crazy, to take the risk to go to competence, what do you have? You have inhibition to take the risk. What do you need in order to do this, to make a decision to take a risk? You have something that will inhibit your inhibition. That's the idea. But you want this thing to inhibit your inhibition according to the context of what the others are doing. Okay? That's the idea. So now we'll go fast. I'll save you all the details. See, and you have some oscillations. Open the window a few times to be able to make the transition or not. That's it. We have implication for this. Zan mentioned at the beginning that we were the first to imprint memories in a Petri dish, neurons in a dish. Many people try to do it. Many people that are much more experienced than us in neuroscience. They try to do it teaching by reward. And I have to tell you one thing about neurons. There are excitatory neurons and inhibitory neurons. They try to do reward to boost up the excitation, or to do it by punishment to boost up the inhibition. What we did, we call it imprinting, teaching by liberation. We inhibit the inhibition. By inhibit the inhibition, we let the network show some new pattern of activity, and you have to do it carefully at a local place and do some first real-time analysis of the activity, and so on and so forth. It sounds always simpler when you say it in words. Okay? But that was a very nice implication of the idea on this neurochip. Okay, how decision? Sometimes you have in a biofilm, you can have more than one type of species of bacteria. Some of them master in keeping information, like resistance to antibiotics. And when the biofilm is exposed to antibiotics, some bacteria need this information. So what happens here is something very interesting. The bacterium that has the information about the resistance to antibiotics, when there is antibiotics, sends signals, whoa, I have the information. So the bacterium that wants the information sends pheromones, and that it, or she, or he, wants the information. And then what happens is that they start to conduct a very long, 15 minutes or so foreplay, because they have to trust each other. She has to trust that he has the information, and he has to trust that if he will give her the information, she will give him food back, or vice versa, replace the she and he. And there will be no transfer of viruses, because what they do after this foreplay, they really form a physical contact between them. You see? They really form a physical contact between them through which there is a transfer of the genetic element that has the information and transfer of food in the opposite direction. So that's the most dangerous sex that you can think about, okay? Because it's directly to the brain. And the brain of human being does not have immune system. Bacteria do have immune system, or version of immune system, and here is an artist concept of the same thing. it's a long time, 15 minutes, so we don't have time to work. This I skip. We were able to quantify the social IQ score of bacteria. We found that our bacteria is here, about 3 standard deviation above the average. Okay, Scientific American liked it. That's a joke here. She talked with me. The reporter asked me, what is interesting here? And I said, well, that the bacteria colonies that we have are not just beautiful, but also smart. She said, oh, so they are not like model in Hollywood. And I said, yes. And said, okay, so I'll start here with this statement. I told her, no, don't start with this statement. But she did anyway. Okay. Applications. New strategy to fight bacteria. When you have 2 sibling colonies that go side by side, what happens? If they are far away, you ask over there how far they can communicate. You see this is 10 centimeter. And you see there is some attraction. So they do sense the other one very, very far away. It's 100 kilometers. If you put them very close to each other, there is a repellent. If you go them and look closer, what you see is that in this here, there is complete inhibition of the growth. Because it's smarter, they don't want to inhibition one into the other, because then they will be confused, lose identity. So we isolate the material from here, show that it really inhibits a colony. You take the material. Again, it's a painful process of about 2 years, and then you see the sequence, compare it to the genome information, make this protein and identify it and so on, and use it, and we discover a new type of toxin that bacteria produce in order to kill themselves. And now you have a new strategy. You don't use something from the outside to control the bacteria. You use something that they themselves use. And that's very efficient because they are unlikely to develop resistance. So this is one application. There is application to fight cancer. Cancer cell, one of the main problems is that after you have the tumor for some time, they start to invade the body. And they make a decision when to start to invade, and if to invade, if to do what they call pathfinder, that they go in the matrix, try to look for a new location, which is usually in the area of the tumor, which is less dangerous, or to make path generator that can really eat the environment, the matrix, and get to the bloodstream. And this is just to show you some simulations that you can understand that from bacteria again, we did the same on bacteria. You see that here it would stack. This maze represents the body matrix, which is complex. You see that here there is a gradient, chemotactic gradient to this direction, and this poor guy stuck. Here what they do, they have a self-assistant mechanism which they secrete all the time. Bacteria do it, ants do it, other animals do it, and which repel it. So if it's stuck at a place, this material accumulates and he tells himself to go out. And you can do then simulations of when to do path finding or path generating. You see they eat the round and so on. Once you realize that cancer has communication and really act as a meta-community, then you realize that the entire approach to our cancer should be different from what we did so far. Yeah.
So if cancer has all this ability to navigate and send out, et cetera, that says that it's really strongly conserved in multicellular organisms that you mostly think of as static. What's conserving the gene set?
Yeah, okay. Excellent question, because we don't understand how can it be that cancer on one hand is a mutation, and on the other hand, it has all these wonderful quotation, sophisticated abilities. Okay, so apparently it might be that in some cases the cancer is really random mutation that eventually enables to do the thing. But in order to be really cancer cell, it has to have all the other changes in the genome. And usually they have 30, 40 different mutations. And they are used then also normal cells, recruit them or enslave them to do jobs that they cannot do anymore. They make decisions and so on. So the entire picture of cancer has to be revised. So we'll stop to do the approach that we do right now, which is stupid because we are not willing to admit that they are smart, which is logically, it's a catch. If they are not smart, how come all the smart people that fight cancer didn't succeed to fight it? Okay, so you can fight cancer with cancer, you can fight cancer with bacteria, you can temper the communication, you can do many other things once you just realize or learn from the bacteria. New robots, I do it very fast. You can show that you use some of the idea, and the minute that you take some agents and add some interaction between them, that if they are very close by, they repel, okay? If they are far away, they attract. If they are at some distance, they align. You can see how you have emergent phenomena in a very simple way. This is agent. See, there is a gradient, and they're going to a target that you don't see. There is a slope here, and you don't see that they go. The minute that you add this simple interaction, yeah, you can see this. You see how they move? No leader, the question that was before. They turn. You'll see in a minute that they all turn around, but there is no leader. They just through the interaction. That's a very simple interaction. And that's very important to many things. Understand navigation of other cells, making new robots, so on. Did you see how they turn around, all of them as if at the same moment? And this is the target. This still wander around. You can do more than that. You realize that if you have the navigation, again, you realize from bacteria what the bacteria do. They coordinate and adapt the interaction. Each bacterium adapts the interaction to the other according to the conditions. Then you can make them even more efficient to move in a complex environment. This group will split, and this group will continue together. Okay, so you can have many more. Because they go together and have these things, sometimes they push themselves out on the heels, but eventually they will do much better. We don't have time, so I'll skip it. This main work of Adi Clark that she received the Google Award. So Google recognized this before. Okay, so this is if you have adaptable interaction, you see the optimal path is much shorter than if you have fixed interaction, and the distribution is much narrower. I'll skip this part. New game theory. Human decisions, emotional or rational. We like to think that they are rational. I'm now convinced more and more that they are emotional. A comment about gut feeling. Gut feeling is real because you have many bacteria. The number of bacteria in your guts is about 10 times the number of cells in your body. The genetic material in the bacteria in your guts far more exceeds the thing in your body. And what you eat and what the bacteria do in your guts affect your brain. And if you eat one type of food, you might make one type of decision. If you eat different type of food, you might make a different decision. Okay? Because it really affects, there is a strong, very complicated communication maybe next time about the brain and our gut bacteria. So let me end with this additional movie. Oh? Make it high, the movie. The decision-making of bacteria tell us that under pressure or under stress, we are likely to make mistakes no matter how intelligent we are. And everything, all I said is a mistake, and let me end here. Thank you.